This lesson is about your muscles and how you are able to move. So some basics on this first bit of notes is the goal. Our goal of our muscular system is to be able to coordinate your muscle movement and control when your muscles are contracting and relaxing. There are three different types of muscle in your body. They're skeletal muscle. Striated means that you can see the lines or the stripes. Those are the myofilaments. Those are protein strands that you can see in the muscle. Skeletal muscles are also voluntary, which means you control when they contract. For example, your bicep. Smooth muscle does not have those stripes that you see right away when you look at the cells. They are not under voluntary control. So an example would be the muscles that make up your stomach. The last type of muscle is cardiac, the muscles of your heart. They do have the protein stripings to them, but of course you do not control it. All right, next off, we have some other connective tissue that is very involved in the muscular system, but it's not a muscle. The first is cartilage. Cartilage is what covers the ends of all of your bones so that you can move your elbow joint and the bones aren't scratching each other. There's cartilage wrapped around the end of each bone. It protects them and it helps keep it flexible. They're also in places obviously like your ears and your nose. All right, the next one are ligaments. A ligament connects a bone to a bone. So like for example, your ACL. It's flexible enough to move your joint. So connecting the two bones, actually three bones in your arm, is a ligament. Lastly, tendons. So usually when I say the word tendon, people think of their Achilles tendon. Oh, I can't get this to move. Here we go. Tendon connects muscle to bone. It's tough. Like if you think about it, your Achilles tendon is quite a hard piece of connective tissue, but it's flexible enough that it can stretch and allow your quadricep muscle to move back and forth. So tough, but also flexible. Example, Achilles. All right, here's a little diagram so you can see what I'm talking about with the striping. Though you might not be able to see it perfectly in here. Smooth muscle does not have the striping. If you look across these cells, you can see the stripes. Those are the protein fibers that make up the microfilaments within it. Both smooth and cardiac are involuntary, and skeletal was voluntary, under your control. All right, this picture shows, here's your bone, and then this is the tendon connecting the muscle to the bone. And if you take the muscle and cut it this way and look at these fibrils, myofibrils are these large bundles that come out. Each one of these would be a cell. So there's 3 muscle cells right here, and within each cell there's a lot of myofibrils. These are packages of protein. I'm going to explain how these proteins work. The individual strands of proteins, like myosin proteins and actin proteins, are called myofilaments. So a filament is single protein strands. Fibrils are the bundles that they come in, and all together those bundles have cells, and the cells make up your overall muscle. All right, here is another picture. This is if I just look at one grouping of myofibrils. So this whole thing would be one cell, and there's 1, 2, 3, 4, 5 myofibrils in it. Because it's a cell that's a muscle, you're going to expect mitochondria. So here's a mitochondria here. There should be a nuclei somewhere around here. We don't see it in this picture. And then instead of ER, they have a lot of SR, sarcoplasmic reticulum. In a muscle, its main job is to store calcium, and calcium is needed for the muscles to contract. So all of this tubing that you see running over the myofibrils is called sarcoplasmic reticulum. Where the tubes join, this is called a T-tubule. T stands for transverse, so it cuts across there. There are thick fibers and thin fibers, and we call the individual protein fibers filaments. So these myofilaments, myo, M-Y-O, always means muscle. These muscle fibers that are thick are called myosin, and actin are the thin ones. All right, so I figured we should do a little drawing ourselves to make sure that you have this in the notes. So we're going to try and draw what we just saw there. So here is our entire cell, and the membrane of the cell is called the sarcolemma. So that's the membrane right here. And we'll draw a nerve coming in, and a nerve branches off with endings. So this would be our motor neuron. And this place right here where the nerve ends and it meets with the muscle, that synapse is called a neuromuscular junction. All right, we can also have some mitochondria along here. And we'll have a nucleus right here. All right, and then I want to show that inside of this cell, when you're looking in it, you can also see all of these tubings. And these tubes, here, here, and here, are called sarcoplasmic reticulum. And I'm going to put calcium inside of them, Ca+2, Ca+2. So you see the calcium in there. And they are united by tubes that run along this way. So this would be called a T-tubule. T stands for transverse. Transverse meaning it cuts along them. So transverse tubules. That'll be important in order to spread the message from the nerve through all of the muscle cell very quickly. And then if I was able to take out just a little, this would be one myofibril. Remember that last diagram had 5 myofibrils in it. The myofibrils are what actually have the protein inside of it. And that's going to be the part that actually contracts. So the second picture I want us to draw is going to be this. A sarcomere is one contracting unit inside a myofibril. So a sarcomere goes from a Z-line to a Z-line, and it has regions where you see dark, thick fibers and regions where you see thin fibers. So if I'm able to zoom out of this, the thick fiber in the middle is the myosin, and the thin ones are actin, connected to the edges. And these are the Z lines because they zigzag back and forth. So we're going to draw that one sarcomere. So we are going to have a Z line here. The distance from Z line to Z line is one sarcomere. So another Z line here, and this to this. Here to here is one sarcomere. So when a sarcomere contracts, that will get smaller. All right, attached to the Z line is actin. And actin, again, is the thin protein. So we're going to have actin coming out here, here, and here, and across on the same side, on the other side, we'll have some more actin. So all of that is actin right there. Actin has a complex wrapped around it. It's called Troponin Tropomyosin. So the tropomyosin wraps around like this. I'll just do it on this side just to show. So this is called the Troponin Tropomyosin complex. That wraps around the actin, sort of prevents it from being attached to. So this Troponin Tropomyosin complex is going to have to move out of the way in order for this muscle to contract. And when this muscle is going to contract, what happens is the middle fiber. So in the middle, this is one way of remembering it. The one in the middle starts with the M. It's also thick. This is called myosin. And myosin has they look like golf club heads. They have little heads on the end of it, little fibers that come up like this. Well, those heads want to grab onto the actin and pull it in. So if I stand here in the middle and I'm myosin, there's Z-line and Z-line, and I'm in the middle, I'm not attached to the Z-line. I have these heads, which are my hands right now, that want to grab onto actin and pull it in. And if you can picture that for a second, the Z lines will pull in, and that is a contraction. So here's my bicep. When it is all stretched out, the Z lines are all far away from each other. And when it contracts, the Z lines all pull in. So a contraction just means the myosin heads have attached to the actin and pulled in, shortening the muscle. So your muscle is long, and then when you flex it, it is pulled in and is much shorter in length. In order to do that, this troponin tropomyosin has to move out of the way, and calcium is what helps that move out of the way. So we're going to go through the steps of how that occurs. All right, we call this mechanism the sliding filament mechanism. Step 1, there has to be a motor neuron telling that muscle, that effector, to contract. So the action potential from the motor neuron arrives at the neuromuscular junction. And that, remember back at our picture, that was where the motor neuron innervates the muscle cell. All right, step 2. Acetylcholine is our neurotransmitter for voluntary skeletal muscles. So acetylcholine is a special neurotransmitter, and that is released and it depolarizes, so it changes the charge. Instead of it being a negative charge inside of the muscle cell, it's going to depolarize it, make it go closer to 0. And our sarcolemma, again, was the membrane of our muscle cell. When it does that, this wave of depolarization, so opening gates, happens throughout the muscle cell, and it spreads through that T-tubule, the transverse tubule that cut through the muscle cell. When it does that, you'll remember there was sarcoplasmic reticulum connecting those T-tubules. So when the sarcoplasmic reticulum is depolarized, the calcium gates open. So sarcoplasmic reticulum stored the calcium. When it's depolarized, it lets the calcium out. And hopefully you're thinking, where's the calcium going to go? It's going to get that green troponin tropomyosin complex out of the way. So calcium binds to troponin, which moves tropomyosin, exposing the binding sites on the actin for the myosin. So again, if I think about this, here I am myosin. I have my myosin heads that want to grab onto binding sites on the actin, but right now it's blocked, so I'm not contracting. If calcium comes flooding into my area, the troponin tropomyosin complex is more attracted to calcium than actin, so they move out of the way. Now the myosin heads can grab on, and when they grab, they actually swivel and pull. And when they swivel and pull, they will shorten the distance between the Z lines. So ATP attached to the myosin allows it to bind. And when it binds, it forms these cross bridges. Cross bridges are when I had my hands and I made that connection. They swivel and they pull the Z lines closer. You need ATP because you are doing an action right there. All right, then to relax. In order to relax, you also need ATP. So the myosin has to release the actin, and that's going to require some ATP. The other thing that absolutely is going to require ATP is we need calcium to go back to the sarcoplasmic reticulum. If you want to stockpile something and put it in just one place, it's going to be going against the gradient. So to move things from low to high concentration, that also requires ATP. So the calcium gets pumped back into the sarcoplasmic reticulum. Sorry, I'm having difficulty moving this screen. Calcium is pumped back into the sarcoplasmic reticulum, allowing tropomyosin to cover the actin binding sites again. Now it's back to normal, and you could have another contraction happen at this point. All right, contractions are graded. So if you think about grades, what does that mean? You can get an A, B, a C, or a D. If I want to flex my muscle and I want an A, or I have to do like a really strong flex to get an A. This is like an F. So your different amounts of flexing that you do show that you actually have control over how much you contract. It's the number of sarcomeres that you are engaging. The more sarcomeres you engage, the stronger of a contraction you have. So It is not like your nervous system. Remember, your nervous system, it either met the threshold and fired that action potential impulse, or if it didn't meet the threshold, it didn't fire. Nerves have what we call an all or nothing response. These have a graded response. Muscle fibers can be stimulated to different degrees. So, lots of stimuli, more myofibrils activated. It's really not letting me move my board up today. So, lots of stimuli, more myofibrils being activated, more sarcomeres contracting. All right, that is the end of today's video. So make sure you look back through your review book and ask me questions. We will be doing a role-playing class and also taking steps like we did with the nervous system and making sure that you can put them in sequential order.